Okay, so now we're going to start with chapter 6, which is about microbial growth. So, microbes in the environment, once they obtain their energy source, they use it for their catabolic reactions, anabolic reactions, and through this anabolism, they can form new cells, like in this case of this Serratia marcescens bacteria that it's undergoing binary fission. So, now, aside of nutrients, microorganisms need other factors to grow, including having the right temperature, the right pH, and the right osmotic environment. So we're going to see the classification of microbes according to their physical requirements. So physical requirements will be temperature, concentration of hydrogen ions, and osmotic pressure. Now the chemical requirements will be all the nutrients that these microorganisms need to grow, including carbon, a source of carbon, nitrogen, sulfur, phosphorus, trace elements, oxygen, and organic growth factors. And of course, we know that all of these atoms, they are used to form plasma membranes, nucleic acids, proteins, lipids, anything that the microorganism requires to make a new cell. So in terms of temperature, temperature for the microorganisms, it is important to grow, and then the temperature will be in a range, and you can have within that range the minimum temperature that the microorganism can use for growing. This is called the minimum growth temperature. And then within that range, the point where the microorganism grows the best, this will be the optimum growth temperature. And then within that range, the highest point in temperature that the microorganism tolerates and can still grow, that will be the maximum growth temperature. And no matter what classification of the microbes you have, it always will be having these types of temperature: the minimum growth temperature, the optimum growth, and the maximum growth. So that classification for these microorganisms in general will be secrophiles for microorganisms that love cold temperatures. Philos means loving. In this case, secro means cold. Meso means middle or moderate. So in this case, mesophiles will be microorganisms that they live in temperate or moderate temperatures. And then thermophiles will be microorganisms that they love high temperatures. They can grow at high temperature. So here is a graph showing you the different microorganisms, and then we have some that are microorganisms that they can love colder temperatures or really, really high temperatures. So we have also other categories besides the three ones that we saw. So these are the psychotrophs, mesophiles, and thermophiles. And this peak in each of these classifications, including the ones that like extreme temperatures, this will be the optimum growth temperature. While the one that it's at the bottom, in the left side from that curve, represents the minimum growth temperature, and the one on the right side at the bottom will be the maximum growth temperature. So for these psychotrophs, we have a range between 0 to 30 degrees Celsius, with a peak around 22 or so. For mesophiles, between 10 to almost 50, peak 37 degrees Celsius. Thermophiles, from 40 to a little bit over 70 degrees Celsius, the range. Optimum growth temperature, like 62. Now, psychrophiles, which is different than the psychotrophs, they have a temperature from range from -8 to 18, with a peak of around 12. Hyperthermophiles from 65 to 110, with a peak of 120. Let me see, let me get my, sorry, like around 95. So we might have a table there that give you more accuracy of those, but so the temperature for growth and the curve, it tells you that metabolically speaking, you can reduce the metabolism at the lowest range, at the lowest point of the range. Like in any of these, like psychrophiles, like close to -10, the psychotroph 0, mesophiles 10, thermophiles 40, the hyperthermophiles 65. So you have inhibited their metabolism to the lowest. The enzymes, they don't grow as much, or sorry, they don't work as much at that temperature. And then in here, of course, in the peak, everything is perfect. The temperature is what it helps them to perform their metabolism at the optimal level. But then when you go to the other extreme, to the highest point, to the maximum growth temperature, and then why the growth decreased? Because that's the rate of growth from 0 to 100%, let's say there. So in here, what it tells you is that you're starting to nature proteins. And it can be structured proteins like channels, like ribosomes, or it can be the enzymes. So here is the actual range, and then when they can grow. So both psychrophiles and psychotrophs, they can grow at 0 degrees Celsius, which is the freezing point of water. And the optimum growth for psychrophiles will be 15 degrees Celsius and between 20 to 30 for the psychotrophs. So this one, the psychrophiles, because their range is actually from, if you see it here, from -8 to 0, the actual minimum temperature. They can live deep in the oceans and in polar regions. And there are scientists that goes to the Arctic, and then they find samples that has these psychrophiles. Then the psychotrophs, these will be the bacteria that they can cause food spoilage, including some mold that they can grow in fruits or in bread. Mesophiles, the optimum growth will be between 25 to 40 with a peak exactly at 37. So these can live in the human body. It's part of the normal microbiota, or it can be the pathogenic bacteria. And actually, most of these mesophiles, in terms of temperature, will be the neutrophiles in terms of pH. So they can grow at the pH of the human body as well, in the case of plasma pH, for instance. So we care about this because if we know the range of temperature where they can grow and we know the minimum temperature and the maximum temperature, we can manipulate this temperature for preparing food or preserving food. So when it comes to food safety, we have something that we call the danger zone. And this thermometer on the left, it's in Fahrenheit, while this one on the right, it's in Celsius. So the danger zone will be, or the danger temperature is where bacteria rapidly grow. And some bacteria not only grow, but produces substances that are toxic for us that we call the toxins. So we have then in degrees Celsius, this danger zone that can be from 12 Celsius to 50 degrees Celsius. Still close to this danger zone, you still have some growth of bacteria. It's very slow, but you want then, if you're preparing food, keep the food away from this danger zone. And then when you're going to preserve it, of course you have to go below this. So then some of these temperatures are basically used to prepare food. Now, let's say you already prepare food and you prepare a large amount of food. What are you going to do to keep it away from this danger zone? Well, if it's a food that undergo the process of heat and then you want to cool it off, well, you might consider to use shallow containers. So let's say here we have a big pot with rice. This rice can grow a bacteria, it's called Bacillus cereus. And then this is a 6 inches depth container, which is 15 centimeters. So if you subdivide this, let's say, into 3 of these 2-inch depth containers, you run the risk of, if you run less risk of growing this bacteria in the danger zone because it takes you less time to cool it off than if you keep your rice in this big, tall container. So this large container will stay within the danger zone at least 5 or more hours, and with this one, you spend it like with 2 hours. And now in the food industry, when they prepare large amounts of food, what they do actually is not only put it in small containers, but sometimes they can have big paddles that you can freeze, and then that one cools off the temperature of the food to prevent you from having problems. So that's all for this beginning of this chapter. I will see you tomorrow.